This is the lecture and explanation for Lab 12, Nitrogen Metabolism. The experiments in this lab are used to help identify unknown bacteria. And for your unknown, you should have already determined the shape of your bacteria and the gram reaction of your bacteria. And in doing so, knowing the shape and the gram reaction, this really reduces the options for the identity of your unknown. Once you have reduced your options for your unknown, then it is time to build the biochemical profile. With the carbohydrate lab, I showed you this figure, which shows a lot of the different pathways for different types of metabolism. And last time we focused on the carbohydrate metabolism. This time we are focusing on amino acid metabolism. So building your biochemical profile means you are determining which biochemical reactions your bacteria can perform. And to do this, we use indicators. So various indicators that determine if certain reactions are going forward or not. Last time in carbohydrate metabolism, we focused on how carbohydrates are used to generate energy. And this shows the glycolysis here going into the Krebs cycle. And of course, the purpose of this ultimately is to produce ATP using the electron transport chain. This lab is going to see how nitrogen metabolism fits into this overall process. Nitrogen metabolism is breaking down amino acids and proteins to help generate energy. And that is usually only done if the carbohydrates are missing. So some of the enzymes you are going to look at, one is gelatinase. Gelatinase breaks down protein, the protein gelatin, into different types of amino acids that can then filter into that process to generate ATP. You will also look at urea hydrolysis. So urea is also part of the cycle that can filter in, break down some amino acids and filter them in to generate ATP. You'll also look at tryptophanase, which breaks down tryptophan, and also cysteine dehydrogenase, which can filter into this process. The first experiment you would have performed in this lab is hydrolysis of gelatin. Gelatin is a protein, and you are determining if your bacteria can break it down, can perform a hydrolytic reaction on the gelatin to break it into the different amino acids. So the enzyme that is involved is gelatinase. And again, ACE, you know that is an enzyme. This is the enzyme that breaks down gelatin. And again, it's going to break down that polypeptide into the individual amino acids. So when that happens, the gelatin is no longer intact, and then the gelatin will not be solid at room temperature. So this is an example of a tube. It's laying on its side, and this is negative for gelatinase. The bacteria that were inoculated, you can see a little bit of the bacteria here that were stabbed along the center. They did not have gelatinase. They were unable to break down the gelatin. So when this is cooled to room temperature, the gelatin remains solid and it does not spread throughout the tube when it's held sideways. If a bacterium is positive for gelatinase, that means the bacteria breaks down the gelatin in the agar, and so that gelatin is no longer solidified at room temperature, and when you tilt it to the side, the gelatin flows down like a liquid. So this is a positive for gelatinase. Negative for gelatinase, the gelatin remains solid. Positive for gelatinase, the gelatin is liquefied. Indole production would be the second experiment that you would have done in this lab, and indole production is looking for the presence of tryptophanase. Tryptophanase, ACE, is an enzyme This is an enzyme that breaks down tryptophan, and tryptophan is a type of amino acid. When tryptophan is broken down by tryptophanase, it produces pyruvic acid and indole. And pyruvic acid, you should remember, is the product of glycolysis. When glucose is broken in half, it produces 2 pyruvic acid molecules. For this experiment, the indicator you would have used is Kovacs reagent. When Kovacs reagent comes in contact with indole, it turns pink. So for this experiment, this is the media you would have started out with. You would have inoculated it, and after 48 hours of incubation, you would have added about 10 drops or so of Kovacs reagent, which is less dense than the media, so it would have remained on the top of the media. And when Kovacs reagent comes in contact with indole, it turns pink. So this is a positive reaction. Negative reaction, the Kovacs reagent would remain that sort of orange, or sometimes it's a green color. Positive reaction, Kovacs reacting with indole, is a pink color. The next experiment you would have done would be hydrogen sulfide production. And for this, the enzyme you are looking at is cysteine desulfurase. So again, an enzyme. Again, this is biochemical profile. You're looking at the enzymes that are produced. And cysteine desulfurase breaks down cysteine. And again, cysteine is a type of amino acid. When it breaks down the cysteine, it causes the release of hydrogen sulfide gas. Then this hydrogen sulfide gas interacts with iron to produce a black precipitate. So in this test, this would have been a deep solidified agar in a test tube, and in that agar there's iron. You would have stabbed with the needle the bacteria all the way to the bottom, and if the bacteria have the enzyme cysteine desulfurase, then hydrogen sulfide gas would have been released, and you would have seen a black precipitate. So this means that is positive for hydrogen sulfide production, positive for cysteine desulfurase. If there's no black color, that is a negative result. The next test would have been urea hydrolysis. Urea is a waste product when proteins are metabolized. So this is a particular waste product produced when cells are forced to use protein or amino acids to generate energy, ATP. Urease, so this is the enzyme, urease is the enzyme that breaks down urea. And when it breaks down urea, it produces ammonia and carbon dioxide. Ammonia, you should know, has a very high pH, so it's a base. So in order to determine if urease has broken down urea, we're using the indicator phenol red. And this is the range for phenol red. Around neutral, it's orangish color. If it becomes basic, if the media becomes basic, it turns yellow, and you use this for the phenol red broths in the carbohydrate lab. But this time we're looking at the production of ammonia, which will raise the pH. So then you're looking for a change in color to pink. It usually ends up a pretty bright pink. So the original media would look like this. It would be a liquid. You would inoculate it with the bacteria, let it incubate for 24 hours. And if ammonia was produced, if urease was able to break down the urea, then you would see a very pink color. So this is a positive, and if it remained that sort of orange color, that is a negative result. The last experiment you would have done is to determine if nitrate and or nitrate reduction are occurring. Now these two processes are a little bit complicated, so you'll want to spend some time trying to understand what's going on. But basically you are looking for the possibility of two different reactions. So the first reaction is nitrate reductase. So this is the enzyme you are determining if it's present. You're trying to figure out if nitrate reductase is able to reduce nitrate, NO3, into nitrite, NO2. So the difference between the two words is the A versus the I. So nitrate reductase reduces nitrate into nitrite. The second reaction you would be looking for is for the presence of nitrite reductase, so that's the enzyme, which can reduce nitrite into ammonia or nitrogen gas. To do this, the two indicators you are using are reagents A and B, nitrate reagents A and B, and together they can detect the presence of nitrite. So that is what they're indicating, the presence of nitrite. You will also possibly be using a zinc catalyst, and what zinc does is it catalyzes the reaction converting nitrate into nitrite. So the two sets of reagents you would be using are reagents A and B first, and then determine on that result, you would determine if you need to use the zinc catalyst. An overall flow chart of what you would be doing is first adding nitrite reagents A and B, and you would look for a change in color. If the media turns red, that means nitrite is present, and then you are done. So what that means is nitrite was present, so this first reaction took place. If instead the media remains that tan beige color, then that means there is no nitrite present, which means either nitrate is still there or the nitrite was further reduced into nitrogen gas or ammonia. So to test for this, you would add the zinc dust. After adding the zinc dust, if it turns red, that means nitrite is present, which means neither of those reactions occurred. Instead, if when you add the zinc dust, it remains tan, that means there is no nitrite, so there's no nitrite present, and that is because it was further reduced into nitrogen gas or ammonia. So that means both nitrate reduction occurred and nitrite reduction occurred. Okay, to go through it again, you are looking for two possible reactions. The first reaction is nitrate being reduced into nitrite. The second possible reaction is nitrite being reduced into nitrogen gas or ammonia. And again, the flow chart, the order of what you would be doing is adding nitrite reagents A and B. If the media turns red, you're done. So if the media turns red, that means nitrite is present, and so that means only this first reaction occurred and nothing else because the nitrite is still present. If instead the media remains that tan color, then that means there is no nitrite present. So your options are that none of the reactions occurred, so the nitrate is still present in the media, or it means both reactions occurred and the nitrite was further reduced to nitrogen gas or ammonia. So to determine this, you add zinc dust. The zinc dust is going to catalyze the nitrate into nitrite if the nitrate is still present. So if it turns red, that means that is what happened. The zinc catalyzed the reaction from nitrate to nitrite. So that means none of the reactions occurred. If instead, when you add the zinc dust, it remains tan or a gray color, then that means there's no nitrate to be converted into nitrite, which means both of those reactions occurred. So as an example, here is a tube of the broth. You added the bacteria, incubated for 48 hours. You've added reagents A and B, and it turns a red color. So that means that nitrite is present. So the nitrite is present, which means all you had was nitrate reduction. On another tube, you add reagents A and B, and it remains beige in color. Then that means you need to add the zinc dust to see what happens. So what that means is that there is no nitrite in that tube. So after you add the zinc dust, it will either remain beige. So if it remains beige, then that means there was no nitrate present, which means, no nitrate present, which means that both reactions occurred. So this is positive for nitrate reduction and positive for nitrite reduction. If instead you add the zinc and it turns red, that means that nitrate was still present. So that means neither reaction occurred. Again, as you build a biochemical profile, first with information from the carbohydrate metabolism lab and then information from the nitrate metabolism lab, you will compile that information and compare to the known profiles. So again, the known profiles, that is in a file on Eagle, and again, the names are over here, but you are looking at the tests. So here you would start from the gelatin liquefaction all the way to the nitrate reduction and compare those results. So this should help identify your unknown.